A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Executive Director of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics relating to antibody discovery and development. We are especially pleased to have Dr. Laura Walker here. In 2021, Dr. Walker was the recipient of the Antibody Society's James S. Huston Antibody Science Talent Award. Jim Huston, the founder of the Antibody Society, was a pioneer and visionary antibody engineer who invented the single-chain variable fragment. The award is given to recognize and encourage upcoming scientists in the field of antibody engineering and therapeutics. Today, Dr. Walker will discuss her insights into the memory B cell responses induced by SARS-CoV-2 exposure based on her work and that of her colleagues. Please note this webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I now turn the show over to our speaker.
Thank you, Janice, and thanks to the Antibody Society for inviting me to speak here today. I'm going to talk about some of the work that my group at Adamab and my group at Adagio has been doing over the past year and a half or so, characterizing B cell responses induced by different types of heterologous SARS-2 exposures. So just to start, this is showing you a timeline of the emergence of SARS-CoV-2 variants of concern, below in red, and also the deployment of COVID-19 vaccines, as shown on the top in green here. And so the point that I want to make on this slide is that really during the first 2 years or almost 2 years of the COVID-19 pandemic, the majority of exposure to SARS-CoV-2 was to the wild type form of the virus or sort of close to wild type forms of the virus. And if you exclude the beta and gamma variants, because they didn't really spread very much globally, beta was essentially restricted to South Africa and gamma to Brazil. So now you're looking during the first year at the Wuhan 1 strain, D614G alpha and delta, which have somewhere between 1 and 2 amino acid substitutions within the receptor binding domain relative to the original Wuhan 1 strain. And then, of course, the vaccines, the COVID-19 vaccines, which were deployed in early 2021, are also based on the original form of the virus, the Wuhan 1 strain. It wasn't really until the emergence of the Omicron variant in December of 2021, I'm showing here BA1, but of course that's been followed by BA2 and 4 and 5, et cetera. But the point is that this was the first major, to use influenza terminology, sort of shift in the virus where we're seeing a very large number of substitutions within the receptor binding domain. I'll talk about that on the next slides of the implications of this. So what we looked at, and this is the first sort of half of my talk today, was how exposure to a very antigenically divergent variant like BA1 sort of modifies the B cell response that was originally established by vaccination within early and with the Wuhan 1 strain, essentially. So that's the first half of the talk. And then in the second half of the talk, I'm going to speak about a different type of heterologous exposure, which is a heterologous prime boost vaccination regimen, where in our study, we were looking at antibody responses in individuals who were primed with the AstraZeneca vaccine, the ChAdOx vaccine, which is based on sort of the native form of the S protein, and then boosted with the Moderna mRNA-1273 vaccine, which is a stabilized form of the S protein, and we asked similar questions. So just to start with this, essentially a summary of the neutralizing antibody response that's induced by the original form of the virus or these very early ancestral forms of the virus. So we and many, many others have shown that the neutralizing antibody response to SARS-CoV-2 is very what's called public or convergent, where the vast majority of individuals induce very similar types of antibodies, which have been classified as these so-called class 1, 2, and 3 antibodies, which have very similar modes of recognition. And these are structures here of just 3 sort of representative antibodies in each class. And these structures were solved by Pam Bjorkman's lab. And these classes are defined, at least in part, based on shared escape mutations. So, for example, many of these class 2 antibodies are sensitive to substitutions at position 484, class 1 antibodies, these substitutions, particularly 417, which has emerged in many variants. And so it's not that surprising when you look at the amino acid substitutions that are present in different variants of concern and variants of interest, which are some of which are shown here on the right, that many of them encode substitutions that negatively impact binding by these common classes of antibodies. On the right here is showing you Omicron BA1 and some of the sublineages. And you can see that this particular variant contains substitutions in the binding sites of all 3 classes of these neutralizing antibodies. And that's why this variant is so immune evasive. And so this is a table from a paper from David Ho's lab, just to sort of illustrate sort of the extent of immune evasion of Omicron BA1 in one of the sublineages, which is shown in the bottom row here. These are representative class 1, 2, and 3 antibodies. Some of them are therapeutic or, EUA authorized antibodies. And you can see that Omicron is evading, almost universally evading these common class 1, 2, and 3 antibodies induced by natural infection or vaccination with the original form of the virus. And that's the reason why when you look at serum neutralizing activity against Omicron in individuals have received 2 doses of an mRNA vaccine, you can see those neutralizing titers are very limited to no detectable neutralization. And this gets better after 3 doses, but it's so much lower than what you see with other variants of concern. And that's, of course, led to significantly reduced vaccine effectiveness even after 3 doses. And that's what drove the massive surge in COVID-19 caseloads in December and January of 2021. And of course, now we're seeing another surge due to a subvariant of Omicron. And so what we decided to look at in our study was the antibody response to Omicron breakthrough infection. This is BA1 breakthrough infection in individuals who had received either 2 or 3 doses of an mRNA vaccine. So 4 of our individuals had only received 2 doses, and they experienced breakthrough infection somewhere between 5 to 11 months after their second shot of the vaccine. And then we recruited 3 individuals who had 3 shots of the vaccine, and they were infected about 1 month after that 3rd shot. We collected the blood samples 14 to 27 days after breakthrough infection, and that's an important point, which is that we're looking at the acute B-cell response here rather than the longer-term B-cell response, which is a follow-up study that we're doing. So we looked at serum antibody, binding and neutralization, memory responses, and characterized monoclonals, and that's what I'll talk about today. So, of course, the first thing we looked at was just serum IgG binding activity, both to BA1 as well as to the wild-type form of the virus. So this is looking at anti-RBD IgG on the left and IgAs on the right. And what you can see is that in individuals who had breakthrough infections, they mounted binding responses to both wild-type and BA1 with only 2-fold lower responses to BA1. Whereas when you look at the vaccinees for comparison, people will receive 2 doses of mRNA or 3 doses. And for the 2-dose donors, we looked at 2 different time points. You can see there's a more dramatic reduction in activity against BA1 as expected based on the data I just showed you. When you look at serum IgA binding, which is shown on the right here, you can see an even more stark difference where in the breakthrough donors, you have similar levels of anti-RBG IgA serum antibodies to both wild-type and BA1, whereas in the mRNA vaccines, you see very little IgA responses, which is not surprising. This is a systemic vaccine, of course, in a mucosal infection. So we also looked at neutralizing activity to BA1 and also other variants and a more divergent SARS virus, so the original 2003 SARS virus, which is shown on the right in green here. And what you can see is that in individuals who have never been infected with any version of the virus and had 2 doses of an mRNA vaccine, they show the highest titers, of course, to the D614G strain, lower titers to these different variants, particularly low titers to BA1 and even lower to SARS-CoV. Whereas in the BA1 breakthrough donors, you see a much broader serum neutralizing antibody response, at least to the variants of concern, where there's a lower drop in activity to BA1 and pretty similar responses to Delta and a little bit lower for Beta. So it seems that infection with BA1 seems to overall broaden the response, at least the serum antibody response. Interestingly, you don't see a boost in serum antibodies to the original SARS virus, which was at least surprising to me, given that the site on the RBD that's recognized by many of the SARS cross-reactive antibodies induced by SARS-CoV-2 infection or vaccination is not mutated in Omicron. But nevertheless, we didn't see a boost in these in SARS neutralizing activity. So we also looked at B cell responses in the breakthrough individuals and compared them to individuals who had only received 2 or 3 doses of mRNA in both the IgG memory compartment as well as the IgA memory compartment. And interesting, as you can see on the left, so this is looking at the percentage of IgG positive B cells that react with the wild type RBD, which is in gray, or the BA1 RBD in green. And you can see that the overall magnitude of the IgG positive memory response, the RBD in the breakthrough individuals, is actually not much higher than what you see in individuals who had just received an mRNA vaccine. So it doesn't seem that breakthrough infection leads to a massive expansion in memory cells, although, of course, we don't have pre-infection samples, so this might represent a large expansion relative to what these individuals had before infection. We don't know that. And on the right, similar to what we saw at the serum and IgA response, you can see that there are IgA memory cells in the breakthrough infection donors to both wild type and BA1, whereas in individuals who had only received mRNA vaccines, you see some response to wild type, but little to no response to the BA1 RBD. So we also looked at the specificity of these B cells, and we did that by staining PBMCs with the wild type form of the RBD in one color and the BA1 RBD in another color. And so you can see with these quadrants here, you can calculate the proportion of B cells that are only binding wild type, that are cross-reactive, and they're only binding to BA1. So in the first graph here on the left, this is looking at the total B cell population as expected in the donors who had experienced BA1 breakthrough infections. You see a larger proportion of B cells are BA1 cross-reactive. And if you look just at activated B cells, so this essentially removes the resting memory cells that were induced by the vaccine and were not re-elicited by the infection, you can see almost all of the B cells in the activated B cells in the breakthrough infection donors are cross-reactive between wild type and BA1. So there are few to no B cells that are only reacting with BA1. And so what this suggests, and I'll show you more data later, is that in breakthrough infection with BA1, sort of reactivates cross-reactive memory cells that were originally induced by the vaccine. So we were also interested in understanding whether or not exposure to BA1 influenced essentially the B cell immunodominance hierarchy. So looking at sort of the proportion of B cells that are reacting to the individual subdomains within spike. And so we looked at the level of B cells using the staining strategy shown on the top, where we gate on all of the B cells that show activity, reactivity with the spike. And then from there, we say what proportion of these spike reactive B cells react with isolated NTD-RBD or S2 subdomains labeled in different colors, and then you can quantify this, which is shown below. So you can see that in the breakthrough individuals, there are a similar proportion of NTD-reactive B cells compared to individuals who had received mRNA vaccines. But interestingly, when you look at the proportion of B cells to the RBD and S2, you can see it's swapped, where there are a larger proportion of B cells reactive to the RBD and BA1 breakthrough donors relative to mRNA-vaccinated donors, and then the other way around for the S2 subunit, which was surprising to us because we were expecting the opposite phenomenon, because of course the Omicron RBD is very divergent from the wild type RBD, whereas the S2 subunit is highly conserved. So you would expect a high frequency of pre-existing cross-reactive S2 specific B cells that should be reactivated when you experience an Omicron breakthrough infection, and the opposite for the RBD. And so our only explanation for this result, and this has also been seen in Gavin Screeton's lab, is that there are a much larger number of, there's a proportion of serum antibodies reactive to S2 that are now binding to the Omicron BA1 S2 subunit, essentially masking that subdomain and preventing those S2 specific B cells from being activated upon re-exposure. Whereas the RBD is more wide open because there are fewer serum antibodies that are able to react with the RBD. And so B cells binding to conserved sites in the RBD are able to be re-elicited. And something similar has been shown in the context of influenza and malaria, this whole idea of serum antibody masking. And so the next thing we did was we isolated monoclonal antibodies from a subset of individuals who had experienced BA1 breakthrough infection. So we selected 5 individuals. This is just showing a pre-pandemic control to show you there's essentially no reactivity with the wild type or BA1 RBD. So we actually mixed the BA1 and wild type RBDs together to just sort all RBD specific B cells in these individuals. And on the right is showing you the proportion of RBD antibodies in each individual that are wild type specific, BA1 wild type cross-reactive, or BA1 specific. So you can see almost all of the monoclonal antibodies we identified were cross-reactive with BA1 and wild type, as we saw at the level of B cells. There are very few BA1 specific antibodies, as I mentioned earlier, indicating, this isn't surprising, but indicating there's not much of a de novo response at this very early time point. It is possible that you'll see a de novo response at later time points, a few months down the line after you get a germinal center reaction going, and we're going to look at that at 5 to 6 months post-infection. So we looked at the sequence features of the BA1 activated B cells, which is shown here. So on the left are clonality plots showing you sort of the level of clonal expansion in these antibodies, and the right is showing the level of somatic mutation. And so as you can see on the left, you can see that the B cell response is relatively diverse. So the big gray slice showing you unique clones. So most donors, and all donors, more than half of the antibodies we identified were unique. So the response doesn't seem to be massively dominated by this big clonal expansion. So that's a little bit different than what you see with influenza, for example, where you see much larger clonal expansions, or in HIV. And on the right is showing you somatic mutation. So you can see that in the BA1 breakthrough donor antibodies are relatively high levels of somatic mutation that are similar to what you see following 3 doses of mRNA or a number of months following primary infection. And so what this indicates is, almost certainly the B cells that we were isolating were originally elicited by the vaccine and now activated by BA1 infection. And further support that when you look at the binding activities of the antibodies to the BA1 RBD versus the wild type RBD, you can see almost all of the antibodies bind to both, with a large proportion of the antibodies binding with higher affinity to the wild type RBD, again, supporting the fact that they were originally elicited and matured towards the Wuhan 1 strain and now re-elicited by BA1. So this is showing a plot of the neutralizing activities of the antibodies. So the proportion of RBD antibodies that show neutralization against the wild type early ancestral form of the virus D614G versus BA1. And so you can see in all donors, over half of the, or near half of the RBD antibodies showed neutralizing activity against D614 and BA1. And as we saw with the binding activity, a large proportion of the antibodies showed more potent activity against the original form of the virus. And interestingly, when you look at these neutralizing antibodies, the ones that neutralize D614G and BA1 shown on the top here, the majority of them are cross-reactive across many different variants of concern, including BA2, beta, and delta. So it seems that BA1 is not only inducing antibodies that are cross-reactive with BA1 and the original virus, but also all of the variants of concern in between. Interestingly, very few of the antibodies cross-neutralize SARS-CoV-1, which is consistent with what we saw at the level of the serum. So there were 2 antibodies we identified, which are shown here, that showed very broad activity against all of the variants of concern that we looked at, and showed some activity as well against SARS-CoV-1. They both compete with ACE2. And we also looked at competition with our clinical stage antibody Adagio 20, of which one competes. So these are 2 potentially promising candidates to move forward into clinical development, either combination with each other or in this case with Adagio 20. So interestingly, when you look at the sequence features of the neutralizing antibodies, much like what I explained at the very beginning, following exposure to the wild type virus, we saw that many of the antibodies had convergent sequence features indicative of a public antibody response. On the left is showing you the breakthrough neutralizing antibodies, and you can see that about 40% or over 40% actually of the neutralizing antibodies utilized one of 4 different VH germline genes, which are shown on the right here. And I'm comparing that with mRNA-vaccinated donor neutralizing antibodies in the baseline repertoire. And so interestingly, in the BA1 breakthrough donors, we found antibodies that utilized the VH353-366 germline gene pair, and many of these antibodies are also induced following mRNA vaccination or infection. So we looked more closely at these antibodies, and much like what you see in these sort of class of antibodies induced by the wild type form of the virus, they have shorter CDRH3s than average, which are shown here. So this is showing you some of the data on the antibodies that we and many others have isolated from individuals who were naturally infected with the Wuhan 1 strain, and you find these in vaccinated individuals as well. And so what you see is after exposure to the wild type form of the virus, when you look at neutralizing antibodies, which are all shown on the left here, a huge proportion of them use the 353 or 366 germline genes. And these are very similar germline genes, which is why I'm grouping them together. And much like the antibodies that use this germline gene pair from breakthrough donors from primary infection, these types of antibodies also use shorter than average CDRH3 lengths. But unlike the antibodies from breakthrough infection, which I just showed you, and I'll show you again on the next slide, these VH353, 366 type antibodies induced by the wild type form of the virus typically do not recognize variants that contain amino acid substitutions at position 417, and some of them don't recognize variants with the E484K substitution as well. So this is different than what we're seeing with the 353-66 antibodies induced by BA1 breakthrough infection. These also appear to be binding to a similar epitope because they compete with class 1 antibodies like Regeneron 10933 and one of our class 4 antibodies. You can see the structures on the right. So they're binding in the same vicinity as these other 353-66 class 1 antibodies. But unlike those, you look below here, these antibodies cross-neutralize BA1, unlike the majority of 353s, 366s that are typically induced by vaccination. And you can see they're broadly cross-reactive among many of these different variants of concern. So it seems that they're binding to an overlapping epitope that's kind of distinct from the kind of classical class 1 353, 366 antibodies. So this is some more information on the other convergent type antibodies that use either the VH169 or the 39 germline gene pairs. The competition data is shown below. So you can see that these many of these VH169 antibodies and 39 antibodies compete with class 3 antibodies, CoV-2 2130, which is one of the antibodies in the AstraZeneca cocktail, Regeneron 10987, which is a class 3 antibody. Obviously, this is one of the antibodies in the Regeneron cocktail, and S309, which is sotrovimab from Vir. So these appear to be binding conserved sites, sort of class 3 sites. And much like the 353 antibodies, you can see that these are also broadly cross-reactive, not just to BA1, but also other variants of concern as well. And so the conclusion, at least to this part, is that the acute B cell response that we've observed following BA1 breakthrough infection appears to be primarily mediated by recall of cross-reactive memory B cells that were originally induced by the vaccine. It seems that breakthrough infection kind of redirects the immunodominance hierarchy from the S2 subunit, which is what you see with mRNA vaccination, where a large proportion of B cells react with S2, toward the RBD. And we think that's potentially due to increased serum antibody masking of the S2 subunit, which is, of course, more conserved. We find that BA1 neutralizing antibodies utilize convergent sequence features, suggesting they're recognizing similar epitopes, and they show broad cross-reactivity with SARS-CoV-2 variants of concern. And so this type of response suggests potentially that future variants might contain substitutions in the binding sites recognized by these antibodies, given that it's a very public response, and these types of antibodies might now be pressuring Omicron out there in nature. And the induction of the very broad responses following breakthrough infection, of course, suggests that booster immunization with a heterologous variant like Omicron might induce similar broadly cross-reactive responses and potentially confer broader protection against future VOCs relative to current vaccines. That's the first part of the talk. The second part of the talk, as I mentioned, I'm going to talk about some of the work that we've done looking at a different type of heterologous exposure, which is this essentially mix and match vaccination, heterologous prime boost vaccination. So there are a number of reasons why you might be interested in heterologous prime boost vaccination, and those are just bulleted at the top. One is to potentially augment vaccine efficacy and durability. Some of these vaccines are more effective than others. You could imagine if you've been immunized with a vaccine that isn't particularly immunogenic, you could come in and get boosted with mRNA, for example. There's also the interchangeability of the vaccines, which might have a sort of operational advantage to address some of the global vaccine supply challenges. Minimization of anti-vector responses, that can be a potential issue with the adenovirus-based vaccines. And then finally, as I'll talk about on the next slide, there's potentially a reduced risk of safety events with a heterologous prime boost vaccination strategy. So there are essentially two different applications that you can think about. One is a heterologous primary vaccination, and that's what we have focused on, where you are immunized with one vaccine and then boosted 3 to 12 weeks later with another. Or you can think about heterologous boosting in a third dose where you get a primary series with one vaccine and then come in many months later with a different type of vaccine. So this is just one slide on the safety issue. So as most of you know, if you read the news, it was reported a number of months back that the adenovirus vaccines, the ChAdOx vaccine from AstraZeneca and the J&J vaccine are associated with very rare but also very serious thrombotic events, mostly in younger women. And so when this was sort of revealed, there were multiple countries that halted the distribution of the AstraZeneca vaccines, at least in certain age groups, and they recommended a switch to a heterologous mRNA booster regimen, either with the Pfizer or Moderna vaccine, despite the fact that there was very limited safety and immunogenicity data at that time. And so we were interested, at least in looking at the immunogenicity and also looking at antibodies induced by this type of regimen. And so we worked with collaborators in Sweden, Matthias Forsell's group, to recruit individual Swedish donors who were between the ages of 28 and 62 years old, all of which had received a ChAdOx1 prime immunizations. There were 55 individuals. And then 65 to 87 days later, half of these individuals were boosted again with the ChAdOx1 vaccine. So that's the homologous boost group. And half of them were boosted with the Moderna vaccine. That's the heterologous boost group. And much like in the BA1 breakthrough study I just showed you, we are looking at the acute B-cell response here. We collected the blood early on, 7 to 9 days following exposure. And we did a similar study where we looked at serum antibody responses, mammary cells, and monoclonal antibodies. So just one note on these vaccines, which I alluded to at the very beginning. The SARS-CoV-2 spike protein is metastable. It exists on the surface of the virus in the pre-fusion form, but it can transition either spontaneously or upon receptor binding to the post-fusion form. And so binding to the receptor triggers shedding of the S1 subunit and a rearrangement of the S2 subunit from the pre-fusion to the post-fusion form. That's a very dramatic rearrangement. So importantly, the AstraZeneca vaccine, the ChAdOx1 vaccine, expresses the native form of the S protein. It's unstabilized. And so what you expect is that cells that are infected with this vaccine express both the pre-fusion form of the spike protein, post-fusion S2, and then you have shedding of the S1 subunit. So you would expect antibody responses to all of these different forms of spike, whereas the Moderna vaccine encodes a pre-fusion stabilized form of S called S2P. So you would expect a more homogeneous expression of pre-fusion S on the surface of cells. And so we're interested in understanding how, if you're primed with this form of spike and you come in with the stabilized form, how does that kind of influence your B-cell response? So this is showing you serum binding activity, IgG binding activity to the spike protein, and then the individual subdomains following either homologous prime and boost, so that's the ChAdOx1 group, or heterologous prime and boost, that's the ChAdOx1 mRNA-1273 boost. So you can see after prime immunization with ChAdOx1, everybody induces a binding response. When you come in again with ChAdOx1 a few months later, so this is the homologous boost group, you can see there is a rise in binding antibody titers, although it's relatively modest, only about 5-fold, and a bit lower for the individual subdomains. Whereas when you come in and boost with the Moderna vaccine, you can see that there's a very dramatic rise in serum binding titers, so about 86-fold improvement in binding titers to spike, for example. We also looked at binding titers to seasonal coronaviruses, HKU1 and OC43, to look at whether or not primer boost immunization was sort of activating pre-existing memory cells that were originally induced by infection with the endemic seasonal beta coronaviruses. And we're comparing binding responses with pre-pandemic samples from other Swedish donors. So you can see that after ChAdOx1 prime immunization in both groups, there appears to be an enhancement in binding activity to both HKU1 and OC43 relative to the pre-pandemic donors, indicating there is likely an expansion of these B cells that were originally induced by seasonal viruses. They're getting expanded by the vaccine. But we do not see any further expansion following either homologous or heterologous booster immunization. This is some data on the neutralizing response, and it essentially mirrors what we saw in the binding responses, which is after prime immunization with ChAdOx1, there was very few individuals mounting much of a neutralizing antibody response, even to the early form of the virus, D614G. After booster immunization, it looks better, although the titers are still relatively modest with an average titer of about 1 in 82. Whereas when you boost with do a heterologous boost with mRNA-1273, you can see a much larger rise in serum neutralizing titers, not just to D614G, but also to other variants. Here we're looking at beta and delta. And this is just quantifying that. And so you can see in the heterologous prime boost group, almost, everybody is mounting a neutralizing response. And even to beta and delta, you can see half or more than half of these individuals have serum neutralizing titers over 1 to 500 to these variants. Whereas in the homologous groups, you can see a large proportion of the individuals have weak to undetectable neutralizing responses to beta and to delta. All right, so we also looked at, as I mentioned, the early B cell response, the plasmablast response. And for those of you that aren't sort of familiar with plasmablast responses, this is just sort of one slide explanation of what that is. So this was first described, or one of the early studies describing this sort of expansion of B cells in peripheral blood following influenza vaccination. So what you can see is after you immunize with the influenza vaccine, which is of course a secondary exposure, 7 days following vaccination, you see this large expansion of plasmablasts. So these are activated B cells that are short-lived in peripheral blood. So these express CD27 and CD38 on the surface. And interestingly, by day 14, you can see this population essentially completely disappears. This is sort of this transient burst of B cells that are actively secreting antibody in the serum. So for flu, plasmablasts average about 6.5% of all circulating B cells 7 days following exposure. So we were interested in looking at this in the context of homologous and heterologous booster immunization. This is quantifying the plasmablast response using those same markers that Patrick Wilson was using in the previous slide, and comparing to pre-pandemic donors. So you can see after prime and boost with ChAdOx1, there's very minimal expansion of plasmablasts in peripheral blood. When you come in with mRNA-1273 as a booster regimen, you can see a larger expansion. So these plasmablasts constitute about 3.3% of total CD19 positive B cells. You can compare that to healthy adults, flu, yellow fever, and dengue. So you can see it's pretty similar what you see with flu and yellow fever, much lower than what you see with dengue, where you see this absolute explosion of plasmablasts in peripheral blood. So we were also interested in quantifying the spike response in terms of the proportion of B cells that were reactive with the stabilized spike and with the recombinant form of S, wild type S. And so recombinant wild type S is essentially a mixture of the prefusion form, postfusion S2, and shed S1 subunit. And so we did this by, much like what we did in the BA1 study. On one color, we had S2P on PE, the P channel. And then on the APC channel, we were staining with this wild type S, which is essentially this mixture. So you can see in vaccinated individuals, you can see a proportion of B cells are wild type specific, wild type S2P cross-reactive. And in this particular individual, in this example, there were no S2P specific B cells. So the first thing we did, we just looked at the overall magnitude of the B cell response, so essentially quantifying spike-specific B cells, whether or not they were, to the wild type form of S or to S2P, we combine them together. And so what you can see on the left is the IgG B cell response, in the middle is IgM, and on the right is IgA. And so much like what we saw with the serum binding and neutralizing activity, you can see after prime and boost with ChAdOx1, you have a larger proportion of spike-specific B cells relative to pre-pandemic donors, but much lower than what you see after booster immunization with the mRNA vaccine. And interestingly, when you look at IgM and IgA responses, you see pretty minimal, frequencies of these types of B cells in homologous boosted group, whereas you see a larger expansion in the heterologous group. And so overall, it seems like heterologous booster immunization is driving a much more robust B cell response. So in terms of the actual specificities, as I mentioned, we use this gating strategy, and we could break things down based on whether or not they're wild type S specific, S2P reactive, or cross-reactive between both forms of the protein. So after ChAdOx1 prime immunization, so that's both of these charts here, so this is before boost. Interestingly, you can see that a large proportion of B cells in most of the donors are only reacting with the wild type form of the spike protein. So they don't react at all with S2P. So that's the kind of salmon colored bars here. After homologous boost, you can see there are still a lot of these wild type specific B cells, whereas in the heterologous groups, the individuals who are boosted with the Moderna vaccine, so this is the S2P vaccine, you can see these wild-type specific B cells essentially disappear, and they're displaced by B cells that are cross-reactive with both S2P and wild-type S. And that's just quantified on the right. And so we asked the question, well, what are these wild-type specific B cells and where are they binding? So we sorted them out and we characterized them. So as expected, all of these antibodies bind to the wild-type form of the spike, but they don't bind the prefusion stabilized spike. Very few of them cross-reacted with the individual subdomains within prefusion S, so the NTD, the RBD, or a prefusion stabilized S2 subunit. Many of them, about half of them, or 40%, cross-reacted with seasonal coronavirus spike proteins, strongly suggesting these are likely to the post-fusion form of S2, just because that's highly conserved among the seasonal viruses. And indeed, many of them do compete with an S2 antibody, just shown below. And you can look at the bottom, look at the levels of somatic hypermutation, which are pretty high in most of the antibodies, indicating they were likely from memory cells versus a de novo response. Importantly, none of these antibodies showed significant neutralizing activity at a concentration of 1 microgram per mil. So it seems that these wild-type specific antibodies are targeting conserved epitopes within S2 that cross-react with the seasonal viruses, but they're exclusively non-neutralizing. So we also were wondering, well, what about responses to prefusion spike? Are these similar following ChAdOx1 boost and mRNA-1273 boost? And so we sorted B cells from a few individuals who had the homologous boost and a few that had heterologous boost, and we sorted out antibodies that were reactive with S2P, either cross-reactive with wild type and S2P or S2P specific. This is looking at the sequence analysis, again, in terms of clonality and somatic hypermutation. So on the top is the homologous group, and on the bottom is the heterologous group. In all cases, you can see that these responses are very diverse. And again, the gray slice represents unique clones. The colored slices represent clonal expansions, with the size of the slice proportional to the level of clonal expansion. So there's a very diverse response. And on the right, you can see that these antibodies are mostly somatically mutated, which is expected because the prime and the boost were spaced about 3 months apart. And you can see those levels of somatic mutation are pretty similar to individuals who had only been infected, and the blood was drawn 3 months later, indicating that sort of the kinetics of affinity maturation following ChAdOx1 prime is quite similar to the kinetics of affinity maturation following natural infection. Interestingly, despite the fact that the homologous and the heterologous boosted groups had similar levels of somatic hypermutation, overall, the antibodies identified from individuals who had been boosted with mRNA-1273 showed higher binding affinities to S2P, and that's likely because the Moderna vaccine expresses S2P, and so you're specifically expanding B cells that react with this form versus the wild type form. But nevertheless, when you look at prefusion S reactive antibodies induced by both of the vaccine regimens, what you can see on the left here is that the vast majority of these antibodies are non-neutralizing. And this is different than what you see with other viruses like HIV or RSV, for example, where binding to the prefusion form of the envelope protein translates into neutralization. If you're binding prefusion, you're neutralizing, and it's really just dependent on affinity. You can see only a small proportion of these antibodies neutralize. And this likely relates to the fact that the recombinant form of the spike protein, even the prefusion stabilized form, is probably not sort of mimicking the native form of the spike expressed on the surface of the virus. As expected, the majority of neutralizing antibodies were binding to the RBD, and most of them competed with the receptor. So this is looking at the proportion of antibodies that are reactive with the different subdomains of the NTD, the RBD, and showing here ACE2 competitive or non-competitive, or the S2 subunit in individuals who had been homologous boosted or heterologous boosted. And you can see overall these immunodominance hierarchies are very similar across these individual donors and across the two different groups. So it seems that at least within the prefusion response to the prefusion form of S, the proportion of the antibodies to each individual subdomain was pretty similar. So we also looked at breadth of reactivity of antibodies from these individuals that had received either homologous or heterologous boosts. So the purple here is shown the homologous group, and in the yellow is the heterologous group. We looked at RBD binding of the RBD antibodies to these different variants, so beta, gamma, delta, lambda, kappa, and Omicron BA1. And interestingly, you can see essentially across the board, the antibodies derived from individuals who were boosted with mRNA-1273 showed higher binding affinities than the antibodies that were induced by homologous boost with ChAdOx1, and similar for the NTD-directed antibodies. And this likely relates to the fact that they were higher binding affinity to the wild type, and that probably translated to all of these different variants. Interestingly, when you look at the Omicron variant, you can see that most of the antibodies are not binding at all, regardless of the booster regimen. And this is just the combined analysis to show you that overall, the mRNA-1273 boost-derived antibodies showed greater breadth of reactivity to variants of concern and interest relative to the homologous group. But the antibodies, most of the antibodies did not recognize Omicron, which is shown here. So this is breaking down the antibodies based on neutralization on the top, with the neutralizing antibodies on the left and the non-neutralizing antibodies on the right. And you can see Omicron is on the bottom here. This is looking at fold change in KD to Omicron versus the Wuhan 1 strain. You can see almost all of the neutralizing antibodies are knocked down or completely knocked out against Omicron BA1. And even among the non-neutralizing antibodies, you can see that many of them fail to react. So the conclusion is that overall, heterologous mRNA booster immunization induces a broader and more potent serum antibody response and memory B cell response relative to homologous boost. We find that a relatively large proportion of ChAdOx1 prime B cells target non-neutralizing epitopes that are not expressed on the prefusion form of S. Heterologous mRNA booster immunization preferentially expands B cells that are reactive with a prefusion stabilized form of S. And overall, they show higher affinities to both the wild type form of the virus as well as to different variants of concern. But importantly, the majority of the antibodies, even the ones that are binding to prefusion stabilized S, induced by both vaccines, are non-neutralizing, and the majority of neutralizing antibodies fail to recognize Omicron BA1, and that's likely to be true on with the sublineages of Omicron as well. And so we think that the development of immunogens that really focus the response on conserved epitopes, either within the RBD or within the S2 subunit, should be a major focus of next generation vaccine design efforts. Most importantly, the acknowledgements of the vast majority of the work that I showed here was done by Kenzie Kaku, who was straight out of college into my group at Adamab and has just been an absolutely brilliant scientist. I want to thank Matthias and Klaas at UMEA for the samples, Margie Agerman at Dartmouth, and Lynn Connolly and Ellie Harshberger at Adagio. And I'm happy to take any questions.
There we go. I'm back. Thank you very much for that very extensive presentation. And we are waiting for the audience to consider the enormous amount of content there and come up with some questions. In the meantime, are there plans to continue to extend this study into the future? Other things that you'd be looking forward to testing? For example, if new vaccines do come along, will you pursue a similar lines of work?
Yeah, so one of the things that we're interested in is the longitudinal response. So as I showed you, there were very few antibodies that were BA1 specific, which is what you would expect in the early response. The question is, when you're infected with a new variant, particularly a very divergent variant, you actually mount a de novo response, essentially recruit naive cells into a germinal center that mature against Omicron. And if you do those cells actually travel to the bone marrow, stay in the bone marrow, and secrete antibody long term? Or are you just boosting wild type immunity essentially every time that you're infected or vaccinated with a new variant. Because of course, if that's just short term, you get cross-reactive antibodies and they last a few months. And as I mentioned, the plasmablasts disappear and then that antibody disappears. If it's not maintained, that's potentially a huge issue, of course, for next generation vaccines, these sort of variant-based vaccines. And I think there are some analogies to, influenza, where you do get, this imprinting phenomenon, original antigenic sin, whatever you want to call it, antigenic seniority, where you're sort of boosting pre-existing memory over and over again with different vaccines. And that's probably what we'll see here with SARS-CoV-2 as well, I think.
We do have a question, sort of a broad overview question, and that is, how do you translate all of your findings for development of an antibody or as therapeutic, so a therapeutic antibody for this purpose?
Yeah, that's a good question. So, as I showed you on one of the slides, some of the antibodies that we've identified from these Omicron breakthrough donors are very broad across all of the variants of concern. Some of them cross-neutralize SARS-CoV-1. So those, you could look at that and say, oh, those are very promising candidates for therapeutic development. But I think you have to consider that the problem with moving antibodies forward as therapeutics that come from patients, in some cases, you could go back and think about the original, that go back to 2020, right? You look at Regeneron, AstraZeneca, for example, Lilly, they identified antibodies from patients. These were these class 1, 2, and 3 type B antibodies. They developed them as therapeutics. And of course, the virus has now escaped those antibodies because they're the same types of antibodies that are pressuring the virus in nature. So, for example, if you move forward with one of these VH353, 366 antibodies, even though they cross-neutralize all the variants of concern, the question is, will they neutralize the next variant? Because you've moved forward with a therapeutic that represents the exact antibodies that are pressuring the virus out there in nature. So the best you can probably do when you think about the RBD is picking epitopes that are not readily targeted by endogenous antibodies. That's one strategy. The strategy we went after with Adagio 20, it was essentially thinking about that combined with the fact that the epitope was very conserved across many more Sarbeca-viruses. But what we saw with Omicron was Omicron is even evading extremely broad antibodies, even some that recognize, Sarbeca-viruses don't even use ACE2 as a receptor. And so going after conserved epitopes within the RBD is not necessarily, that doesn't necessarily mean that you're going to be escape-proof, basically. I think if you're going to go after the RBD, you need a combination of, 2 antibodies, maybe 3 antibodies targeting epitopes that are immunorecessive, and ideally also conserved, because, maybe then they would also be more functionally constrained, at least more so than these very variable sites. And then, of course, there's the S2 subunit, which, as I mentioned, is very conserved. There have been antibodies identified to particular site in S2 called the stem helix that are extremely broad. Some of them even cross-neutralize MERS. The sort of trade-off with those antibodies, and you see this with influenza antibodies as well, is that there's a trade-off between breadth and potency. Those antibodies are very broad, but the potencies are relatively modest. So the best ones I've seen of IC50s around 0.1 microgram per mil. That's 50 to 100 times worse than the top RBD antibodies that you can find, for example, the Regeneron type antibodies or our antibody Adagio 20. And so then you're looking at a dose problem where you might have to give a couple grams to get to the neutralizing titer you need, assuming those antibodies are not kind of punching above their weight in terms of how well they're protecting relative to neutralization. So that's the trade-off. You go with the RBD, you have to worry more about escape, but you have potency. You go with S2, you worry about potency, but less about escape. And so it's a little bit of a conundrum.
Thanks very much.
That was a very long answer to the question. Sorry.
But complete. Thank you very much. We do have a couple of more questions here, and one coming in from Dennis Burton. Thank you. What is wrong, quote unquote, with the S2P conformations such that so many of the non-neutralizing antibodies bind to the protein?
I do not know the answer to that question, but I think that there is something non-native about it. I think it's hard to say what that is, whether or not it's sort of more open than the native form of the protein, for example, so the antibodies can sort of access their epitopes, whereas they can't access them on the native form. maybe it's differences in glycosylation. I think that's a very difficult question to answer, but I think it's likely that it isn't sort of faithfully mimicking the native, trimer on the surface of the virus.
Next question. To get a breadth of coverage, a multiplexed mRNA vaccine, such as wild type, Delta, Omicron, would that be a strategy to go for?
I think that it depends also if you're immunizing or naive individuals or immune individuals, but I think if you immunized with a wild type Delta Omicron vaccine, for example, and you have wild type immunity, you're going to essentially be expanding a lot of B cells, because you have the wild type in there that are just wild type specific, maybe a proportion that are cross-reactive with Omicron. But I don't know if that strategy would be any better. I think it'd probably be worse than just coming in with Omicron, where you're specifically boosting the cross-reactive cells versus the ones that are wild type specific. I think the bigger problem is with the longevity of the response. If you're just boosting memory and none of those, if none of those plasmablasts go to the bone marrow and are maintained in the bone marrow, then the protection is likely to be extremely short-lived, a few months. And I think that's likely what's going to happen, or at least that's what you see with flu, where the cells are not long-lived after boosting.
Next question requires a little bit of speculation. What do you think is going to happen with the variant-specific vaccines that are being developed?
I have no idea. I know there are studies in monkeys by, Bob Cedar and others showing that the neutralizing responses to wild type and different variants are pretty similar if you come in with the wild type boost versus an Omicron boost. So I'm not sure if right now there's a ton of justification for moving forward with these variant vaccines. And then, of course, again, as I mentioned, you have the longevity problem, where even if you do that, if the protection is very short-lived, is it sort of worth it or not? But I'm not sure what's going to happen there.
Next question, somewhat off topic. What are the dominant phenotypic consequences of the high number of mutations on virus infection profile?
I don't know what that means. The high number of mutations in the virus around the antibodies. Can you read it again? The phenotypic consequence of a high number of mutation on virus infection profile. I don't know what that. I don't know. I don't really understand what they're asking. I'm sorry.
Perhaps the attendee could expand on the question. We will give this person a quick chance to do so. But the viruses, of course, many of them do have many mutations, an enormous number of mutations. That is correct. We've kind of lost track. Oh, here we go. On the virus, what are the consequences of escape mutations? Sorry, lost again, Ray. Sorry, man.
They're trying to ask if you have a large number of, I don't know, I'll just try to answer, is that, as we've shown, even with a large number of mutations, there are enough antibodies induced by the original form of the virus, in this case, the Wuhan 1 strain and the vaccine, that, when you come in with a divergent variant like Omicron, you're still able to boost these cross-reactive B cells, many of which are neutralizing, which is a good thing. And John Weary and Michel Nussensweig and others have shown that after 2 doses of the vaccine, and even more so after 3 doses of the vaccine, if you just look at memory cells, about 40 to 60% of them do show cross-reactivity with Omicron. So I think maybe the virus will jump into an animal reservoir and we'll see an even more divergent virus pop up in the future that is evading, all antibodies induced by the vaccine or infection. That's possible. But with Omicron, which is the most divergent variant we've seen to date, there's still enough cross-reactivity that even with 3 shots of the vaccine, you're at least maintaining protection against severe disease, which is a good thing. But we'll see what the future holds there. I think that's what they're trying to ask there, the consequences of escape.
Another question. Okay, let's see here. Why do you think there is a general association between lower potency antibodies and conserved epitopes?
Well, in the case of influenza, HIV, and SARS-CoV-2, I think the problem is with accessibility. So when you look at flu stem antibodies, for example, if you look at binding of those antibodies to just a recombinant stem, they're high affinity, but because the HAs are clustered so densely on the surface of the virus, the antibodies can't access those epitopes. In the case of the stem helix, that epitope is cryptic. It's not always exposed. It's much better after receptor binding. And so I think that's essentially what's limiting potency for those S2 type epitopes. Within the RBD, I think it might be sort of a similar phenomenon when you think about the sort of up-down hinge-like motion that the RBD undergoes, where some of these class 4 antibodies, for example, just can't access those sites. They're actually very high affinity to the isolated RBD, but presumably those sites are not well exposed in the native form of the virus because of the up-down movement.
Okay, well, thank you very much for that. I think we have come to the end of the questions, and I'll draw out saying that just to give somebody a last chance. We are at the end of the questions. So in concluding, I would like to thank Dr. Walker for relating her insights into this memory B-cell response coming from the different types of SARS-CoV-2 exposure. I want to thank the attendees for joining the webinar today. An on-demand version will be available in a few days. I'll send a link to it by email to everyone who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient for you. Thanks again and have a great rest of your day.